This screencast will take you through the figures and the references that you'll need at minimum in your paper. First, for figures, you'll probably want to have a figure of your blast results. So if these are your blast results, make sure to capture an image of your results. So in this case, I'm just going to highlight this area or screen cap it basically. Take that figure, and there you go. I'll take that, save it, and import it into my document. Another figure that you'll probably want to have is going to be the alignment of the nucleotide sequences. Now, here is a muscle alignment of the full nucleotide sequences, both wild type and mutant. But this is probably more than you'll need. This is not especially useful to a reader. The area that is important is right here. So you'll probably want to do is delete all of the above and all of this below to specifically highlight the area where the mutation is. Now, to make this figure complete, just like the previous one, you'll have to name it, give it a figure number, so figure, in this case, perhaps it would be figure 2. Give it a title or a caption of some sort, and this would be a cluster alignment using muscle of the wild type and mutant TNI3 sequences. And in this case, these are mRNA or cDNA sequences, however you want to call them. I guess cDNA is more accurate. You'll also want to include numbers. Now, I deleted a bunch of sequence. This is actually a nucleotide number here, and if you remember that these are 60 across, and you count the number of columns or rows that you deleted, you can figure out the number that would go here, the number that would go at the end of these rows as well. Okay, so this would be another figure that you'd probably want to have in your paper. Another figure that you'll want is the protein alignment. So same thing down here. So this would end up being probably figure 3 or 4 or 5, depending on how many other things that you'd be using. This would be a protein alignment using cluster O, cluster omega, of, again, both wild type and mutant TNNI3 protein sequences, or amino acid sequences slightly more accurately. Now, these are 60 across, so I'm deleting the first 120 just to make this more readable. And let's just shrink this down. So this would be 120, would be the first amino acid in each of these. And the last one in both of these would be, let's see, 120. I'd have to count it out, and I really don't feel like doing that now. So you can do that yourselves to figure out what the last amino acid would be in your sequence in the appropriate area. And actually, I can just delete this because this is the only area that matters in terms of the mutation. So this will be 120 to 180 in both cases. Okay, so this would be another figure you'd need. Another one you'd need would be your structure. Now, I'm going to go back to CN3D, and here we go. It would be good to have images of your structure, at least one. You're probably going to want multiple views that show the various arrangements and interactions. You might actually have multiple structures that you're looking at as well for yours. So you'll want to have an unhighlighted region, but then you're going to also want to take an image that has specifically the area mutated highlighted, so one can compare, a reader can compare the wild type version and the wild type with the area that would have been mutated highlighted. To get an image out of CN3D, there is an export PNG listing here, and you can then convert to whatever file type is necessary after the fact. Okay, so this is a subset of the figures you'd need. You'd also want to have a number of figures as needed to describe the function of the protein, pull these images from other papers, but attribute them appropriately. Lastly, there are a number of references you're going to need for your sequence that you've pulled, the BLAST algorithm, the muscle and cluster algorithms, as well as any other tools that you've used. So first, for your gene sequence, or in this case, cDNA sequence, there are a number of References listed here. The earliest one, which you'd find down here, is the one that is um, worthy of citation. This is the first group that cloned and sequenced this particular gene, and so they deserve the most credit. Um, You can probably find this information in UniProt or OMIM or other sources. This is just the first location where you would have found it. Other things you'll need, a reference for BLAST. BLAST itself, here's the BLAST homepage, was written by people and they need to be appropriately cited. If you go to the help page, you can eventually figure out references here. I'm not going to walk you through all of these things. Same thing for the muscle algorithm for alignment. If you go to help and documentation, or the clustral omega alignment algorithm under help and documentation, you can find it yourself. The translate tool is a little more difficult to cite. I'll let you figure that out, assuming that XPayC, this resource portal, is made by the Swiss Institute of Bioinformatics, and they would be the institutional author. Figure it out from there. Other things you'll need to cite, CN3D, which is the group, the software for looking at structures, but also the group that determined the X-ray crystallography structure of that protein. Also, if you used any other resources, for example, if you used information from OMIM that was not directly attributed to another reference, or information from UniProt as well, you'll want to reference these databases as well. All right, so those are the figures and some of the referencing that you'd need for the writing of your paper.